Hello, everyone. My name is Seth Lundell. I'm a master's candidate at the University of Saskatchewan, and I'm going to be presenting to you guys today on the physiological and gene expression effects of two bacterial inoculants on the salinity tolerance of a 4th-generation alfalfa breeding line. Okay, so here's an overview of my presentation. I'm going to give you guys, first off, some background on soil salinity and context for that, a bit of information on the response of alfalfa to soil salinity, and my research question, as well as my hypothesis and objectives, my experimental design, my materials and methods, information on my data collection and the PCR study I'm hoping to do, as well as a timeline for this experiment and a conclusion. And there will be space at the end for questions. So a little bit of background on soil salinity. As you are all well aware, the challenge for us in the next 30 years is going to be feeding a growing global population on a fixed land base. Soil salinization is a problem because it's eating away at that land base. Globally right now, soil salinity affects 397 million hectares. Specifically in Western Canada, 6 million hectares of arable land are saline, which means that this is costing producers money from lost yields and is also reducing the actual production of those areas, which is a problem because it's taking away from global food production. And this soil salinity is generally caused by the evaporation of salt-laden groundwater on the surface of the soil, which leaves behind those salts, and those can build up over time until they cause issues for plant growth. So a little bit of background on alfalfa. I'm sure you're all familiar with it. It's a deep-rooted perennial forage crop. It's a legume, which means that it normally associates with rhizobium, which are a bacteria which can fix nitrogen and exchange it for fixed carbon from the plant. Alfalfa is grown on approximately 4 million hectares in Canada. It's salt sensitive, above 8 decisiemens of salt per meter, which is moderately saline tolerant. And alfalfa is a staple for livestock. So yeah, it's a very, very important forage crop. A little bit of background on soil microbes, which are also going to play a part in this experiment. Soil microbes can have positive, neutral, or negative effects on plant growth. We're just going to be focusing on the positive ones in this experiment. Two in particular that we're going to be using are plant growth promoting rhizobacteria, as well as rhizobium, which we just talked about. And like I just said, they can provide benefits to the plant in exchange for fixed carbon. A little note here that's also something of importance is that the host plant, so in this case alfalfa, can prevent mutualism in periods of high salinity stress. And that is because, as you remember, these two organisms trade with each other. So the bacteria gets fixed carbon and the plant gets some sort of nutrient. So let's say nitrogen. However, in periods of high stress, the plant actually needs all of that fixed carbon for its own metabolism and its own survival. So it decides not to give any of that to the soil bacteria, and yeah, it can kind of axe the symbiotic relationship. However, the relationship may be strengthened by using soil bacteria which have higher salinity tolerance, but that's not for sure. So anyways, back to the original points here. These microbes can increase plant stress tolerance by improving soil health, by giving plant the access to nutrients, by fixing nitrogen, and also, interestingly, by inducing the expression of plant stress-related genes. So increasing alfalfa salinity tolerance. There are many ways to do this, but the two that we're going to be focusing on in this experiment are like conventional genetic improvement through phenotypic recurrent selection. So we're going to be getting all of our alfalfa seeds from a breeding program that's breeding for salinity tolerance. And secondly, through the use of different bacterial inoculants, which should induce salt stress related genes, as well as fixed nitrogen. So the genes that we're hoping to target are osmolytes or osmoprotectant, genes, and we're going to get into that in a sec here. So my research question: can a strain of rhizobium and of PGPR, so that's those plant growth promoting rhizobacteria, induce increased expression of salt stress related genes in alfalfa, and can this induction be magnified over the course of a multi-generation breeding program? So specific alfalfa stress responses that we're going to be looking at in this experiment. I've already mentioned osmoprotectants. So obviously those are created in response to osmotic stress. And so when alfalfa grows in saline soil, over time, soil salts can accumulate around the roots, which can create water potential, which is not favorable towards the plant. And so the plant can really struggle to take up water. And also when salts are actually taken up into the plant, they can draw water out of the plant's cells. And that can cause a loss of cell turgor, which is not healthy. And so in response to this, the plant creates these osmoprotectants. A couple examples are proline, trehalose, glycine, betaine, and those accumulate in the cells and they act to draw the water back in. So they kind of equalize or create an equilibrium between the salt and the osmoprotectant and keep the cell healthy and happy. So another stress is oxidative stress. So when the plant experiences like drought stress, it closes its stomata to prevent water being lost. This unfortunately also means that the plant cannot take in carbon dioxide to fix carbon with. So the photosystems experience a bit of stress because they're still accepting a bunch of energy from the sun, which they normally direct into carbon fixation, but they can't. And so this energy is leaked into surrounding oxygen molecules, and it creates reactive oxygen species, which are very damaging to the plant cell. They can they can oxidize and damage DNA, proteins, and lipids, and eventually lead to cell death. So the production of antioxidants by the plant combats this. And one of those osmoprotectants we just looked at, proline, actually has antioxidative properties. So lastly, we're going to look at symbiotic stress. So like we talked about, soil salts can reduce nodulation. They can damage the root hairs, which cause or create the nodules initially. Salts can also reduce the effectiveness of nitrogen fixing enzymes, so leg hemoglobin, which creates an environment for nitrogen fixation, as well as nitrogenase, which actually fixes the nitrogen. So the plant can combat this by producing all of these compounds we just talked about, because as it happens, they all increase the salinity tolerance of not only alfalfa, but also of bacteria, as well as increasing the production of leg hemoglobin, which makes a more favorable atmosphere for nitrogen fixation. So my hypothesis is that these two strains of bacteria will increase the production of all these compounds, which will increase the cell tolerance of alfalfa compared to controls. Also predicting that when you pair these bacteria together, they will interact to produce more of these compounds than would be expected by just the sum of the parts. And lastly, in predicting that the production of these compounds will increase along the progression of alfalfa generations in this breeding program. So my objectives for this program are to assess whether these two bacterial strains can confer salinity tolerance to alfalfa compared to controls, to assess through PCR analysis whether recurrent selection, so this breeding program, can increase the expression of these genes for these three osmoprotectants and leg hemoglobin, and finally to assess whether these improved alfalfa or later alfalfa generations can interact with these soil bacteria to be more mutualistic than earlier generations, creating more healthier and larger plants. So the experimental design, it's going to be a factorial randomized complete block design, 5 bacterial treatments that are listed here, 3 alfalfa generations. I know my title says 4, but COVID has kind of slowed things down. So hopefully we're going to get the 4th in time for the second replication of the experiment. And this is kind of the breakdown of how many pots there's going to be. And like I just mentioned, there's going to be 2 experimental replications. Yeah, when we start the experiment out, there's going to be a gradual increase in salinity concentrations to avoid salt shock to the plant because we don't want to kill them outright. And harvest is going to occur 90 days after planting, which is about the time of alfalfa maturity. So some of the indicators of plant success that we're going to be measuring in the interim as well as at harvest are going to be chlorophyll content, nitrogen content, seedling survival, root shoot biomass, plant height, leaf injury scores, and nodulation. So these are going to measure a couple aspects of plant growth. So chlorophyll and nitrogen, the nitrogen fixation, nodulation is that symbiotic relationship, and then all the rest here are kind of just general indicators of plant health. Indicators of salinity tolerance are all these compounds that we've been talking about, as well as like hemoglobin, because they all protect against salt damage. And we're going to measure them all at harvest by these methods here. The PCR study. So as you can imagine, the concentration of all of these compounds, proline, glycine, betaine, trehalose, and like hemoglobin, depends on how much is created in these synthesis pathways and how much is degraded in these catabolic pathways. So if you have a lot that's produced and not very much that's degraded, then you have a lot of these compounds which increase the plant's resilience or resistance to salinity stress. So yeah, these are all the genes that we're going to be looking at in the PCR study. We're going to be extracting RNA from the roots, the shoots, and the nodules at the time of harvest. Through reverse transcription, we're going to see the concentration of the transcripts for each of these genes, and hopefully we're going to find that plants that were inoculated with bacteria, as well as later generations in this program, have more of these transcripts of the synthesis pathway, and hopefully less of the transcripts for the catabolic pathways than either controls or earlier generations. So here is a quick timeline of the experiment so that you can kind of imagine what's going on. This is really, really basic. It's going to be a little more complicated than this, but at the moment, we are culturing 2 bacterial strains for inoculation. I got COVID a week ago, so that's kind of slowed things down a little bit. We're going to inoculate 225 alfalfa plants, which is the first replication of the experiment. Pretty quick here. We're going to grow that alfalfa for 90 days, collect data on those indicators we just looked at. We're going to harvest those. We're going to begin the PCR study. And while we begin the PCR study, we're also going to begin the second replication of the experiment. And then we're going to analyze the results. So in conclusion, this experiment is testing the efficacy of both plant breeding as well as the inoculation by salt or by bacterial inoculants for improving the salinity tolerance of alfalfa. And we're hoping to find whether meshing those two methods together can increase or create a system which is more efficient than the sum of its parts. So thank you for your time. And the floor is now open for questions. Thanks.